In this lecture, we will take a closer look at the prokaryotic cells. Remember that the main thing that sets prokaryotes apart from eukaryotes is their lack of a nucleus. They still have DNA, still have that genetic material, but it's not confined within a separate membrane-bound structure. So when we look at prokaryotes, they have cytoplasm, they have ribosomes, which are the sites of protein synthesis. They have the genetic material, the DNA, the RNA. They have chromosomes, but they don't have that nucleus. They don't have that membrane-bound nucleus. Prokaryotes tend to have a single circular chromosome located in what we refer to as the nucleoid region. So we don't call it a nucleus because it doesn't have a membrane on it or around it. We call it a nucleoid. Okay, so similar to a nucleus, but not quite. Eukaryotic cells also contain membrane-bound organelles that we don't see. So in terms of complexity, the eukaryotic cells are a lot more complex than prokaryotic cells. So what we're going to focus on in this video are the characteristics of the prokaryotic cells. So let me bring up this image here. And this is just kind of our standard boilerplate prokaryotic cell drawing. Okay, so this is going to be our representative bacterial cell. And just keep in mind, just like representative plant cells or representative animal cells, not all bacteria look like this. In fact, very few bacteria would actually look like this. But we can use this model, this drawing, to help us identify different structures and describe those structures. So when we look at this drawing here, we're going to see several things. If we start on the inside, the first thing we're going to notice is that nucleoid region. Okay, so we've got this chromosome here contained within this central region of the cell, and so this would be the genetic material. Again, we call that the nucleoid because there's no membrane around it. Okay. Then we see all of this yellowish material here. That would be the cytoplasm. That's the jelly-like substance that makes up the inside of the cell. And we'll see all of these little dots here. Okay, so we'll see all of these little red dots. Those would be ribosomes. Ribosomes are the sites of protein synthesis. So the DNA, the genetic material, contains the blueprints or the instructions for making proteins, and then the actual site of assembly of those proteins is the ribosome. And then we see these kind of larger spherical structures in here. These are referred to as inclusions. Some bacterial cells have inclusions, some don't. Inclusions can contain any number of substances that can be used for storage by the cell. We'll talk about some of these a little bit later on. We talk about specific bacteria. But for now, just keep in mind that inclusions can be substances that need to be stored in the cell for whatever reason. Then we have this little structure right down here, this little circular purple structure. This is called a plasmid. Plasmids are extra pieces of DNA, not part of the original chromosome. You see that it's not contained within the original chromosome here. Plasmids contain extra pieces of information. Some plasmids contain information for antibiotic resistance. Some contain information for conjugation, for allowing bacterial cells to exchange DNA. And we'll talk more about those later on. But some bacterial cells, we'll see these these plasmids, these extra pieces of information. Now, when we look on the outside structure, we'll see the plasma membrane, and then we'll see the cell wall. All bacterial cells, with very few exceptions, have a plasma membrane and a cell wall. And then we see this green structure called the capsule. Now, in my video for Lab 3, I talked a little bit about the capsule. Not all bacterial cells will have a capsule. It provides some extra protection from the immune system and may allow a bacterial cell to be pathogenic that would not otherwise be pathogenic if it didn't have that capsule. Then we have these other structures. We have Fembree, we have a pilus here, we have a flagellum here. Fembree tend to be shorter projections, often allowing the cell to attach to other cells or attach to surfaces. I kind of think of the Fembree almost like Velcro, that it allows the cells to be able to stick to structures. We have this pilus here, can be used for different things. A lot of times what we see with the pilus is it's used for conjugation. It's used for allowing bacterial cells to swap DNA. And then, of course, we have, in this case, a single monotrichous flagellum here. And flagella are used for movement. You know, bacteria don't have legs. They don't have any way to move around in their environment unless they have these flagella. So the flagellum serves as the form of locomotion for the bacterial cell. In my video for Lab 3, I talked about cell morphologies and different cell arrangements. So I'm going to skip through this section of the textbook, but just know that it's here if you need to go back and review that. So a little bit about the cell wall. The morphology or the shape, the structure of the bacterial cell is maintained by the cell wall. It's found in almost all prokaryotes. There are a few exceptions. We also see the cell wall in some of our eukaryotes, like I mentioned before. But it is on the outside of the cell membrane, and it protects the cell from changes in osmotic pressure. So osmotic pressure is when we have a difference in concentration of solutes on either side of a semipermeable membrane, like a cell. When we have a situation where we have differences in the solutes, water is allowed to pass back and forth across that membrane. Most substances can't. Very tiny substances can. But for the most part, we're just looking at moving water. So as water moves back and forth across this membrane, then it sets up a difference, a concentration difference, this gradient difference. And if we have too much water that is diffused or osmosed, okay, the diffusion of water is called osmosis, then it can cause extreme osmotic pressure on a cell if those dissolved substances changed. So let's take a look at this figure here. We have three solutions. We have an isotonic solution, a hypertonic solution, and a hypotonic solution. Okay, iso means the same or equal. So in an isotonic solution, the solution inside the cell is the same as the solution outside of the cell. So you see here we've got a 20% solute concentration, 80% water. Water flows in and out of the cell in equal amounts. So water is just going to flow back and forth across the cell membrane, cell wall, and we maintain cell shape. So ideally, we want our cells to be in this isotonic environment. There's no net movement of water. There's no net movement of the solutes. But if we deviate from an isotonic solution into a hypertonic or hypotonic, then we can start to have problems. So in a hypertonic solution, the solution has a higher solute concentration. So inside our cell, we're still at 20% solute, 80% water. But outside, now we have 40% solute and 60% water. Water is going to flow out of the cell. It's going to go where there is a higher solute concentration, so water flows out of the cell. As water starts flowing out of the cell, it leads to something called crenation. The cell essentially becomes dehydrated. Now, in a hypotonic solution, so we've still got our 20% solute concentration inside the cell, but now outside of the cell we have a 10% solute concentration. So this concentration is hypotonic. It has less of a solute concentration. So water, again, flowing to where there's a higher concentration of solutes, goes into the cell. This is going to continue until we get to an equilibrium or the cell explodes. It lyses, it ruptures. Okay. So we need this condition of isotonicity. Now, the cell wall helps to protect this to some degree. Our cells, our eukaryotic cells, human cells, don't have a cell wall. So we have no protection really against these changes in osmotic pressure. But for cells that have a cell wall, there is some protection. And that's what's shown here in this figure. In an isotonic solution, the cell membrane pretty much comes right up to the cell wall. We have no net movement of water. Water just flows in and out at will. But when we get to a hypertonic solution, the cell starts to, or the cell membrane starts to kind of fall away from the cell wall. In a hypotonic solution, the cell wall kind of counteracts that to help prevent swelling. So when we have cells that have a cell wall, they are in effect protected from some of the effects of osmotic pressure. Now, that's not to say that this cell here wouldn't eventually shrivel and potentially die, or that if enough water flows in here, then it can overcome the cell wall and explode, but it allows the cell to survive changes in shape longer. Okay, so the cell wall protects the bacterial cell against those changes in osmotic pressure. Okay, so it's very important structure. Now I've talked about the nucleoid region. Here is a scanning electron micrograph where you can see they've kind of outlined this nucleoid region. Keep in mind it does not have a separate membrane around it, but the DNA, the chromosome, is all there together in that central region. Here's a little bit more information about inclusions. Like I mentioned, inclusions were for storing certain substances, glycogen or starches, volutin, PHB. So here are some different inclusions that can be found within prokaryotes. Okay. I talked about endospores in my video for Lab 3, that endospores allow the bacteria to be able to withstand some pretty drastic changing environmental conditions. They can form these spores when conditions are not favorable for them to be alive, essentially, and be growing and metabolizing. And we'll talk more about those endospores a little bit later on. So if we look at the structure of the plasma membrane, we're going to see that the structure of the plasma membrane is going to be very similar regardless of whether we're talking about bacteria or fungi or plants or animal cells. We follow this fluid mosaic model with a phospholipid bilayer with carbohydrates and proteins stuck in it.